anyhow, Alekh is a postdoc in Erica Korb's lab and has done some really interesting work that you're going to hear about, linking changes in epigenetics. So those are the kinds of proteins that control our DNA, and how that might change and lead to autism in the face of endocrine disruptors.
All right, morning everyone. So yeah, like Becky said, I'm gonna be taking more of a molecular approach in like my research. So starting off a lot of molecular before we go into more of the environmental side. But my work is really focused, or my work over the last couple of years has been really focused in trying to understand endocrine disrupting chemical exposure in the context of epigenetics and phenotypic consequences in mice. Started off focusing on one endocrine disrupting chemical, but really branched out into this idea of low dose mixture and the sort of effects that that may have, as that's what we're more generally exposed to. I definitely don't have to tell this audience about endocrine disrupting chemicals, so it's going to be very brief, but as I'm sure all of you know, EDCs affect a wide variety of tissues and cellular systems, and EDCs interfere with many different biological pathways within these cells, including signaling pathways, as well as epigenetic functions such as what's going on in the nucleus, things like gene expression, the chromatin landscape, and just general regulation of genes that control nearly every biological process in those cells. The big problem, again, preaching the choir here, the big problem with EDC is there's a wide range, a huge umbrella of what is contained within EDCs, and the actual molecular consequence of EDC exposure in general is not very well, not very well understood, let alone in combinations that we're normally exposed to. The complexity is only made worse when we bring in the idea of neurodevelopmental disorders, or NDDs, which are the primary focus of our lab. So NDDs are incredibly heterogeneous, incredibly complicated. And one of the most complicated ones for sure is autism spectrum disorder, or ASD. So ASD encompasses a wide range of neurological differences between different people, and more severe forms can lead to things like more severe disorders leading to nonverbal patients that are nonverbal, et cetera. While ASD can be explained primarily by the genetics, it can't fully be explained by genetic or epigenetic functions. And there are several different factors that contribute or influence the severity incidence of ASD within communities. And those include many, there we go, environmental factors. So it's things like the gut microbiota, maybe infections, but also environmental toxicants, including EDCs. Like I said, our lab is primarily focused on trying to understand the epigenetics of ASD, as there are a number of chromatin modifiers, so proteins that are involved in controlling gene regulation in the chromatin landscape, that are known to be mutated or have variants in people with ASD. It's a pretty large group of these chromatin modifiers, so we're trying to understand what is the connection between epigenetics and aberrant neurodevelopment, particularly in the context of ASD. And that's really where my work, especially working with the folks at the Center of Excellence in Environmental Toxicology, brought in this additional angle of environmental toxicology and just how do endocrine disrupting chemicals and exposures contribute to ASD and neurodevelopmental disorder incidence and severity. As there's a growing body of literature that has shown that environmental toxicant exposure, of course, is associated with aberrant neurodevelopment, particularly in low-income communities or communities that are more exposed to a lot of these toxicants. But additionally, there is another growing body of literature that has shown that exposure to many of these toxicants, such as BPA or PFAS, do cause epigenetic perturbations. Many changes in gene regulation, many changes in the chromatin or DNA landscape. Many folks at Penn and CHOP have studied these exact effects, such as changes in DNA methylation following BPA exposure in several different tissues. So that really gets like the overlying, or the underlying, I guess I should say, foundation of my work, or like the question I'm really trying to address. This idea that the incidence and severity of NDDs is associated with not just the epigenetic or genetic aspects, but also these environmental factors. And maybe it is this gene-environment interaction that may be explaining this heterogeneity in things like ASD. Like, do we get more severe, or do patients get more severe phenotypes because they have the genetic component as well as environmental exposures? But before I can go into like the gene environment stuff, I really have to understand the environmental side first, because I'm definitely not an environmental toxicologist by training. I'm a gene expression epigenetics guy. So when I first started this project about 2 years ago, we started from like very straightforward, what are the effects of BPA exposure in our neurological systems, like our in vitro and in vivo models? But in working with folks and extensive collaboration with folks at CEAT, we really got clued into this idea of EDC mixtures, or toxicant mixtures. And this idea that we as people and just across all society aren't just exposed to just BPA, we're exposed to many different EDCs in combination and mixtures at very low doses, well below the no adverse effect level. And so there's very little research that actually tried to understand what is this normal transient level of exposure actually doing to just like neurobiology, especially in communities that may have even higher amounts of it, for example. And that's where we got clued into this paper here by Andrea Gore's team, where she tried to study what is the neurological effects of a low-dose mixture of EDCs in a rat model. So she developed this mixture called Neuramix, which are 9 endocrine disrupting chemicals at doses well below the no adverse effects level, representative of normal human exposure levels as found in serum as well as some placental studies. She put together this mixture and then fed rats during pregnancy, in a small window during pregnancy, just this mixture at the low dose, and then observed what are the resulting changes in the pups that are born. So it's a perinatal exposure paradigm. And what she found is that Neuramix exposure did affect in a sex-specific manner the development of these rats. Some behavioral phenotypes are actually similar behavioral phenotypes that are seen in some neurodevelopmental models of rats, as well as gene expression in the amygdala of these rats. So this provided a really good foundation for the work I could pull forward. What if I use a similar mixture in a mouse model that we use pretty extensively in the lab, and just take it a step further for more intensive molecular, behavioral, and developmental profiling. So that's where I get to my mixture that we'll be talking about for the remainder of this talk, which I'm terming low mix. Low mix is a truncated form of Neuramix. So Neuramix had 9 EDCs. We brought it down to just these 6. Here are the doses that were in the Neuramix paper. I'm just using them again. We chose these 6 in particular as they're, of course, some of these EDCs with the highest environmental health concern, but also these have pretty extensive literature across the board for their association with neurodevelopmental disorders or just association with neurological phenotypes in general during exposure or in communities that are exposed. But also for a number of these like BPA, PBDE, and PFOS, there are several papers that have shown their association with epigenetic perturbations. Like I said, BPA exposure affecting DNA methylation. There's papers that show the PFOS exposure in certain tissues affects gene expression pretty broadly. So we just thought this was a really good truncated list of EDCs to use for a mixture and move forward into a mouse model compared to the initial rat model used in Neuramix. Before we could go into the rats or the mouse, specifically in an in vivo system, we wanted to start with an in vitro system. So we in the lab have this pretty clean system where we can generate cortical neuron culture, like cortical neurons in culture, and do whatever sort of perturbations we want on them to study what are the effects on the epigenetic or gene expression landscape, the transcriptomic landscape, following things like depleting certain proteins, or in this case, exposure. So we dissect out embryonic day 16.5 or embryonic 16.5 cortical neurons and then culture these cortical neurons just like we do any other cell culture. Highly pure system, very much enriched for excitatory neurons, split into 2 groups, where one group we exposed to a dilution of our low mix. Then after 10 days, so somewhere relatively late, moderate time scale into development of these, we performed RNA sequencing just to examine what are the changes in gene expression following low mix exposure compared to just vehicle exposure. And what we found is that there is a pretty modest change in gene expression in these cultured cortical neurons. We see a fair number of genes that are both differentially downregulated in blue, upregulated in red. But the question then with all RNA sequencing is it this paradigm just affecting genes at random, or are there certain classes or families of genes or genes that code certain proteins that may be affected particularly by EDC exposure? So I did what everyone does with RNA-seq and just ran gene ontology analysis, which will look at enrichment for certain categories of genes. And this is what is known as SynGO. So this is a type of gene ontology analysis where the only terms utilized are those involved in synaptic processes. So the cellular compartment specifically at synapses, or proteins at the synapse, or biological processes of the synapse. My gene ontology analysis of just the downregulated genes, I guess I should say that there was no real enrichment amongst the upregulated. Amongst downregulated genes, we did see that there is a very specific downregulation of genes encoding proteins involved in localizing to the synapse, both the pre and the post synapse, as well as an enrichment for these genes that involve an actual synaptic function, particularly things like functioning at the synaptic membrane, as well as some proteins involved in specialization of the synapse. So this is really cool. Even in a mixture form at very, very low doses, especially after the dilution, in a pure cultured system, we're able to see these changes in genes that are really important in synaptic function. So we could use this to jump off into an in vivo model. Now, instead of using a cultured cortical neuron system, what if we instead perinatally expose mice to this low mix in their diet, trying best to encapsulate how we as humans are normally exposed, like pregnant women may be normally exposed. Like I said, with Neuromix, what they had done in that paper is that they took their Neuromix solution and got the rats pregnant, and during a small window of about 10 days during pregnancy, they fed the rats once a day with a wafer with the solution in it. We wanted to go one step further than this because pregnant women don't just eat like a wafer of BPA once a day. They are consistently exposed to this low-dose mixtures of EDCs in their environment. So we instead worked with a company in Vigo to develop an actual physical, like solid food diet that contains either the vehicle or the low mix, and then put our mice on that diet. Well, I should say just the timeline up here. After the mice were equilibrated to our mouse facilities, we placed the mice on the diets 2 weeks prior to mating, and then kept them on the diet through pregnancy all the way up until their pups were weaned. So the pups were also kept on the diet, just in their cages, only access to the food they had available. And then during that development of their actual pups, we tracked certain milestones like weight gain, how quickly or when they were able to open their eyes, when they started start walking around, and other things like tactile startle response, like usual reflexes. After weaning them, we did a circuit of behavioral tests just to examine things like, are there deficits in anxiety phenotypes or social phenotypes? So the top two are anxiety phenotypes, and then this tests social deficits, which are often studied in ASD mouse models, for example, we do in the lab. I don't have time to talk about the behavioral paradigms just for how much time I have in this talk, but I should note that there are some sex-specific differences here. What I really wanted to highlight for this slide in particular were the really cool results that came out of our developmental milestone testing. For the most part, the low mix exposed mice had pretty much the same, they reached the same milestones as their their vehicle exposed counterparts, like open eyes around the same time, their grasp, most of their reflexes were about the same. But the notable exceptions are those color I don't think sticks out really well here. But low mix exposed mice actually develop fur around a day faster than their vehicle exposed counterparts, and up to 3 days, or they develop a tactile startle up to 3 days faster than their counterparts. These were especially cool results because I'm sure many of you know that there is the idea of expedited puberty, especially in cases of environmental exposures, is a growing field of research. In addition, tactile startle, where you just puff some air on a mouse and see when it has an exaggerated startle response and when they actually start developing that response, is indicative of the sort of sensory, hypersensitivity. And there's a lot of cases, or many, many cases of patients or people with ASD that have this sort of tactile hypersensitivity. So the fact that we can see these even with this very low dose over a long period of time perinatally does at least provide some credence to this idea that environmental exposures to these mixtures, these EDCs, may in some way be paralleling the sort of phenotypes we see in people with neurodevelopmental disorders. And then when I get to the gene environment stuff at the end, this idea that what the combination of this genetic factor that results in some of these in addition to the exposure may result in these exaggerated phenotypes. So like I said, I'm a transcriptomic guy, so the part that I was most excited about was now going into the transcriptomics. We did all the behavioral and the developmental stuff, but now we wanted to look at, like we did in the neurons, what are the changes in gene expression following low mix exposure? So after the behavioral circuit, we sacrificed the mice, dissected out the prefrontal cortices, and then performed this highly multiplex single nuclei sequencing protocol called SplitSeq, which is basically, as I'm sure those of you know, with single cell, you usually use droplet-based methods. SplitSeq is a 96-well plate method, and it's really effective at doing all of your samples all at once, all in-house, in a very cost-effective manner. So this allowed us to do 4 mice per condition, so both the vehicle exposed as well as the low mix exposed, and also get both males and females in there to examine these sex-specific differences at the transcriptomic level, just like we have with the behavioral and developmental data. So I'm going to be talking about this for the next couple of slides. So this is just the indicator of what these 4 conditions are: female and male, vehicle, and exposure, these acronyms here. And SplitSeq works beautifully to generate the single nuclei data. So here's a UMAP distributing all of the different nuclei we got. This is just all of our samples all compiled together, mostly to illustrate that in the prefrontal cortex, this methodology allowed us to capture all of the major cell types we expect in the prefrontal cortex. So in the tannish colors, you get all of the cortical layer excitatory neurons. In the purplish colors, you get the inhibitory neuron populations. And then in blue, we get all these non-neuronal cell types with things like oligodendrocytes, astrocytes, and microglia, which I'm particularly interested in because that's very understudied, the effects of ADC exposure in general on these non-neuronal glial cell types. So I'm not going to talk too much about it, but there's some interesting results that came out of that in particular compared to just the neuronal system. All right, so with single cell nuclei data, again, for those of you who worked with it, there's so many things you could possibly do with it. So definitely don't have the time to go into everything, but I'm just going to focus on two bits here. The first one is a very straightforward analysis. Pairwise comparisons of exposed mice versus vehicle exposed mice, stratified by each of the different cell subpopulations we can get. So here is my differential gene expression analysis, or DEG analysis, and excitatory neurons. And I wanted to use this slide particularly to highlight for those of you who are doing single nuclei or single cell or going to do it. There's a growing debate or discussion in the field about this idea that default methodologies for differential gene expression analysis in single cell have a really bad false positive issue. So people who, or a lot of groups, myself included in papers that I published, have the issue of having way too many differentially expressed genes if you just use the standard methods. So there's this growing need to use multiple different algorithms and just examine what genes come out of both algorithms or multiple algorithms and apply some pretty stringent thresholds. So that's more or less what this is showing here, is that I did the DEG analysis by two methods, masked as well as pseudobulk. And I'm showing you just like the volcano plots you normally see of significance versus fold change. And then this one is fold change versus percent expression, which I think is also a really important metric people should be showing more in single nuclei, because the idea of percent expression is how much of a cluster, like all excitatory neurons, actually express the gene. Because if something has a 5-fold change, but it's only expressed in 2 cells, that's far less meaningful than something expressed in every single cell and also has a 5-fold change. So this was just my really intensive way of getting to the genes I'm very confident were differentially expressed following low-mix exposure. And we see that there's a pretty fair handful that come out from both these methodologies. What was especially cool, and will be highlighted in the next slide in particular, is some of these genes are very important in neuronal activity, as well as the neuronal adhesion, or the synaptic adhesion, or the genes that are localized to the synapses and involved in maintaining these sort of neuron-to-neuron connections. So even though there's only a couple of genes that came out here, at least in the non-sex stratified method here, there are still some that are quite important. And then, like I said in the previous slide, the only other group that really showed a couple of these DEGs were actually in astrocytes. So really cool analysis there. That's not just a neuronal phenotype we're seeing from exposure. It could also be in these non-neuronal cells that I'm very interested in drilling more into. The last thing I'll talk about then is just even more intensive analysis of single nuclei or single cell data, where we can examine in RNA sequencing single nuclei differences in single genes changing, but the brain is a highly interconnected system of neurons and non-neurons, and it's really important to understand cell-cell signaling pathways and receptor ligand pairs between these different cell types. And we can actually dissect that using this really cool methodology called cell chat, which will examine changing in gene expression of partners within signaling groups, such as like cholesterol signaling, between cells between each of your cluster. And then examine, are there differences from each of your conditions in signaling pathways instead of just individual genes? So what I initially did was just look at across all known signaling pathways that exist, are there differences in the number of these pathways, or just what is known as the cumulative interaction strength, which is just a normalized metric of the pathways plus their just general gene expression changes. And what's a little disheartening was that if we examine the vehicle, it's kind of very small here, but vehicle in gray and then exposure in green, we didn't really see any difference overall in the numbers or weight. We decided to drill in further, and that's where we actually got the really interesting result. So this heat map here is showing from any cluster on the, it's probably going to illustrate this a little better, from any cluster on the left, so these inhibitory cells, excitatory cells, and non-neuronal cells at the bottom, what are the changes in signaling from that cluster to any of the other clusters, including themselves, noted on the bottom here? So what I'm highlighting here is one of the really cool things we found, where we actually saw that our exposed mice actually had an increase in signaling, just the number of signaling as well as number of the weight of signaling from inhibitory neurons to nearly all other neurons in the brain or the prefrontal cortex. Likewise, we saw that there was a reduction in the signaling from these non-neuronal cells, like the oligodendrocytes I'm noting down here, to a number of the cortical layer cells. So this result we found up here, where it's sort of balanced out, is mostly because there's just a broad dysregulation of signaling from non-neuronal to neuronal cells that sort of balanced itself out. And really interested to try and understand what are the different pathways that may be contributing to these phenotypes. And one of the ones that really stuck out was that cholesterol signaling is one of these that were increased from the inhibitory to the other cell types. So this is a Corey diagram showing just a cumulative weight of the cholesterol signaling pathway in vehicle exposure versus regular, like low mix exposure. And we see that cholesterol signaling is actually increased, particularly in cortical layer cells following exposure compared to vehicle. There's a couple other pathways, again, I don't have time to show, but it is really interesting that this cholesterol signaling in particular is very important in neurobiology. So this one sticking out as one of the top ones was very important. I don't know anything about cholesterol signaling, so don't ask me any questions about it. All right, so finally, like I said at the very top, the main focus of why I jumped into this project in the first place was examining gene-environment interactions. All the things I talked about right now, which are currently in review, was just to really get to like what is Lomix doing in the first place before we could jump into the more complicated stuff. So now I've been starting to work on this gene-environment interaction question. As like I said, big focus on the lab and on my projects is on these chromatin modifiers associated with ASD. We have another paper in review that showed that if you have haploinsufficiency for a number of these proteins that have known variants in people with ASD, they seem to converge on similar types of pathways and genes when you look at transcriptomically, and they affect neuronal functioning. But the thing is that a lot of patients or people with variants exhibit wildly different phenotypes, and maybe it's the environmental exposures that explain a lot of this heterogeneity. Maybe it's a combination of these variants in addition to exposures that exacerbate the phenotypes. So I started off doing the in vitro system again in cultured neurons, examining, like I said a couple slides ago, the exposure results in some change in gene expression. If we were to deplete or emulate haploinsufficiency in some of these chromatin modifiers, we also see modest changes in gene expression. But when you actually combine exposure with the depletion of the haploinsufficiency, we not only see substantially more differentially expressed genes, but all the ones in color here, which is very hard to see, but the colored ones here are unique to the double condition that weren't seen in either condition alone. So there is a very specific, unique, and exacerbated phenotype in the combination compared to either exposure or the genetic aspect alone. And I'm really interested in now moving into the in vivo system, utilizing my perinatal Lomix timeline with a mouse haploinsufficient for some of these modifiers, and then examining, do we see more severe behavioral or developmental phenotypes? And with that, here are my acknowledgements, all the many different funding sources, and I'll take any questions.